Okay, you ready, Katie? Okay, so Katie Wagner is a high school senior duly enrolled at Wayne School of Engineering and Wayne Community College. She will complete her Associates in Science this spring and earn a Wayne Community College Honors Program certificate. In addition, she's an active member of the North Carolina Scholars of Global Distinction Program. Katie is interested in pursuing a higher degree in international studies or political science after graduation, and her talk is entitled Albizia, julibrissin flowering patterns in the United States using iNaturalist. So take it away, Katie.
Thank you. Let me share my screen. Can you see? Okay. I did a presentation of the Albizia julibrissin flowering pattern, which is based for scientific. So you can basically select your research.
Oh, sorry, Katie, you're breaking up.
You know, they can submit photographs, videos, documentation, any sort of biological and questions. Something I find a lot of in North Carolina, and nowadays I think more of it because it's that time of year shopping and blossoming. Background, trees, mimosas, but original
Katie? About. Sorry, we can't. You're breaking up a lot. Are the rest of you guys having that problem? Yeah, we can't. Your voice is breaking up, so there's some microphone problem. I'm sorry.
Maybe try plugging in the microphone again. Just double check your connection.
Katie, maybe you can stop sharing your screen for just a moment.
Yes, we still see your screen. Oh. Uh-oh. Katie, you hear us? You hear us? No.
Obviously, the university wants us to reject Zoom forever.
I know. I think this is like, we're done with this. We need to be doing these things in person. It's very convenient until it's not. But if she continues to have a problem, we can move on to our third presenter and then move back so we don't lose too much time. We'll see if when she comes back, things improve.
Dr. Muich, where can I access this recording afterwards?
So we'll be posting everything to YouTube and we'll send out information once everything's posted.
Hey, Katie, welcome back. Let's
Hi,
try.
I'm sorry. Can you hear me now?
Yeah, totally.
All right.
Yeah.
I had
Go
no
for
idea
it.
what was going
Yeah,
on.
we
but
hear
couldn't
I don't
hear
know.
or see
Yeah, but you
anything.
sound perfect. So go
Awesome.
ahead.
Okay.
Okay.
All right. Play. Okay, cool. So have we gotten here yet? Okay, so commonly known as silk trees or mimosas, these are really common in North Carolina, so that was part of my interest in them. They were originally imported to attract pollinators like hummingbirds, bees, butterflies, and to decorate ornamental gardens. However, even though they are quite beautiful, they do have quite active seeds when they spread, easily spread by weather, animals, that sort of stuff. And when they release neurotoxins when ingested. So in the agricultural South, such as North Carolina, where they thrive especially well, it's really dangerous to the agricultural based factor of our economy. So that's why it's important to figure out what is the flowering period of the Albizia julibrissin and how does that change based on location within the United States. So my original hypothesis was that it would mimic the conditions of the countries in Asia, such as India, Japan, China, where it originally came from. So it would have a midsummer flowering period. And based off of my research, I was able to collect 385 observations from iNaturalist, all of which were research grade, which meant they had a photo, a geolocation associated with the observation, and it was verified by at least 3 experts on the website. So I used search words Albizia, julibrissin, and Persian silk tree to find my observations. And they were found across a decade from July 2011 to November 2020. So it was a wide berth of observations to choose from. A lot of them did not have observations, particularly in the north, which aligned with my hypothesis that it does not do too well in colder climates. But I was able to find that my hypothesis was largely supported with 37% of observations in July and over 80% in the 3 summer months from June to August. So based off of these numbers, which I totaled in the bottom row, all 385 of them are right down there, I found that only 2 of them fell after September, which was pretty surprising, honestly, because you would think that it would get hotter as it moved on. So that was the cutoff for, when the temperatures would be too hot for them to blossom. And I believe that would be when we would most need to be concerned about the seeds spreading and pollinating and continuing to grow as an invasive species. So here's the map that I took from the iNaturalist website, and you can see that the concentration of observations were largely in the south and along the east coast where they were originally imported. And that does tie into this next graph, which you see here. It's a graph that I made myself based off of a website called MapChart. And based off of the observations I saw, as it got later into the year, the color of the states faded, and those without any observations had a green color. Only these was Indiana, and as you can see with both of them side by side, Indiana only had 1 observation, and that fell in July. So obviously it didn't line up very well with the rest of my research, but you could see that largely as the climate or as the temperatures were colder, the flowering period was later in the year. Well, based on my research from other sources and not just from observations I found from citizen contributions, North Carolina State University said that the flowering period for the Persian silk tree fell from May to July, whereas the Native Plant Society in North Carolina said it fell from July to August, which meant my observations fell pretty squarely in the middle, with the average being July. found that was outside of North Carolina specifically was the United States. And it said that it began in May and, oh, I'm sorry. Control. Anyways, sorry. The United States Department, it says that the flowering began in May and continued throughout the summer and through August. So that kind of aligned with my observations as well. So seeing side by side, specifically in North Carolina, it's clustered largely in city areas where there are a lot of animals, a lot of traffic to get the seeds moving around. Compared to the other states, North Carolina did have quite a few observations. So why is this important and how can we build on this in the future? So I found that if we were to build off of my observations, it would be most beneficial to include dates and years. That way we could see how not only how it moves throughout the country and throughout time and during the year, but also between different years, compare how the Persian silk tree has migrated. And by including to expand more countries, on a more global scale, it would be beneficial to include observations around the globe, include other countries and track the year that way as well. So not only would we be able to see how the Persian silk tree changes in location, but also in flowering period, that would tell us, number one, where they are to prevent them from moving around since they are an invasive species. But number two, it would also help us figure out how the climate itself is changing. So we know that the Persian silk tree, it flowers whenever the weather is, the warmest, the humidity is the highest, that it gets the most sunlight. And by seeing how that changes between the years, we would be able to check, the progression of global warming and climate change even. And by expanding to overseas observations, that would be an even more beneficial research development. So in addition to including that, we would protect native wildlife by containing the seeds and monitor for climate change by tracking where it goes throughout the years. So that's why this was really, really important because it has so many substantial impacts. By following it throughout time, we would be able to not only protect the living, but also non-living parts of our environment. And this was significantly, I would say, less lab intensive compared to Sophia's project, but it was also done the fall semester before last. So in the height of the pandemic, and working on a citizen-based research platform, it's really showed me how we can all come together virtually and still find such incredible scientific discoveries using just virtual online contributions through the internet. And I was able to put this project together based on my own online research without having any hands-on experience because we were isolated for such a long time. So I did enjoy being able to still get involved in the environmental community, get to learn more about the Persian silk tree, even though I was not able to, unfortunately, go into a lab.
Okay, well, thank you very much. Yay. Do you want to unshare your screen and then we can see each other? Thank you.
Do you have any idea how I?
Is it on the top? Does it stay on the? There we go.
Sorry.
So many buttons. Yeah. Katie, that was really awesome. I mean, and that is a great project to do when you're not able to be on campus somewhere. So you did a really nice job and you made use of some really nice resources and made some nice graphics and all that. So that's great. Okay. So does anybody have any questions for Katie? And I can start us off. So do does it make seed pods, the mimosa? It makes, and do they fall to the ground? Or
so they actually make legumes, like kind of like beans, so those are the things that usually spread after they come before the flower, or sorry, they come after the flowering. So it's kind of an indicator for when the transition's gonna start. But yeah, they come in little pods.
Okay, and do animals actually eat them? I know you said they were toxic, but do animals
They
actually
do.
try to?
Yeah.
Yeah, that's unfortunate.
I'll ask a question.
Yeah, go ahead.
Those trees are so pretty. I just thought I'd comment. But I was wondering, so they're an invasive species, and I guess people were planting them for a while, and that's how they were spread. Are they still used, sort of like sold and planted by gardeners at this point?
They are actually, because I don't think it's very widely known that they are invasive because they're so pretty. It's, like I said, used as an ornamental decoration for a lot of gardens. We've actually, my own neighborhood has at least 5 just spreading, just sitting there. They keep putting in, I think every other year we get another one, and I'm not sure anybody's actually aware that they are invasive.
Yeah, thanks. Some of the ones I've seen, they seem to be kind of randomly located, like they grew there wild,
I have another question. Oh, Rebecca, did you have a question?
I did. This is, so I'm not a scientist. So my question is more about your process. I appreciate what you said at the end there about having to change your research plan because of the pandemic and what was available to you and, working in person. So has this changed where you see your own trajectory going as a scientist? Do you think you want to kind of branch into a more traditional lab-based community, or do you want to kind of follow this community-based approach?
Oh, that's a good question. I think it's definitely contributed to my interest in taking more advantage of the resources that I have, seeing as I didn't even realize that that iNaturalist even existed until it was, my best friend, through this project, it was my greatest resource. So I definitely learned that I needed that the internet, the web, there are a lot of resources on there for any field, honestly, not just biology, that you can take advantage of. And I definitely know that they exist now because of the pandemic.
Anybody
I
else?
had a quick question, Katie.
Yeah, go ahead.
You may have already said in the beginning, I'm not sure when you had a connectivity issue, but I was just curious, how did you get interested in this particular species?
Well, like I said, they're really common. I don't want to say that. They're really common around where I live. And growing up, we had one about a block away from my house. We used to always climb in it and, swing from the branches, that sort of stuff. So it's a positive memory in my personal childhood. And I think, honestly, that's the main reason. But it's also, there's such a pretty plant. And I hate to say it like that, but it's like, seeing the aesthetics of it, I was like, oh, that would make such a great presentation. And then whenever I had the opportunity to dive into it more, I realized how interesting it actually was. So it's like, it all worked out well.
Okay, great, thank you.
Okay, thank you. So yeah, that's really interesting, just to kind of look around at what's around you. I mean, there's so many interesting things that you could study that are just things that you've grown up with or around you. Very cool. Yeah. All right. Well, if there aren't any other questions, we will move on to our last presentation. So thank you, Katie. Okay, and Paige, you're going to be up. So Paige Turner is a senior pursuing a biology major with a Spanish minor at UNCG with both disciplinary and international honors in the Lloyd International Honors College. She has assisted on more than 6 research projects in the Kerner Biodiversity Lab and School of Education under the mentorship of Dr. Sally Kerner and Dr. Sarah Heredia. So her talk is entitled Impact of Burn Frequency on Arthropod Communities in North Carolina Longleaf Pine Savannas. So take it away, Paige.
Alrighty. The obligatory struggle to share my screen. Hold on. Okay. Do you see the title page?
Totally do, yeah.
Okay. So the impact of burn frequency on arthropod communities in North Carolina longleaf pine savannas. My study takes place in the North Carolina longleaf pine savanna, which is a fire-dependent ecosystem. This means fire is required to limit the intrusion of invasive hardwoods and woody shrubs that would otherwise dominate the understory. Over 1,000 species have been recorded in this ecosystem, including the longleaf pine tree, which are adapted to fire and require it for the release of nutrients from litter and dying plant matter. Deforestation, agriculture, habitat fragmentation, and fire suppression all threaten this ecosystem's ability to persist. At 3% of its original range of 92 million acres, the conservation of the remaining savanna is critical for the 900 species of plants that don't exist anywhere else in the world. Arthropod diversity is essential for the health of the savanna because they perform many roles. Herbivores have a positive influence on above ground plant biomass. Detritivores break down litter and debris between burn periods, and many compose the bulk of the diet for other inhabitants of the savanna. Understanding what influences the arthropod community helps conservationists gain better insights to the limits of biodiversity in the longleaf pine savanna. Fire suppression and burn frequency can shift community dynamics. Lack of fire allows for the intrusion of invasive species. Frequent fires prevent species from recovering between burns, and more than 3 years between burns can lead to the buildup of dead plant matter, which blocks sunlight and stalls circulation of nutrients in this ecosystem. Plant species composition, which is altered by burn frequency, also impacts the diversity of arthropods, as many have evolved alongside select species and will leave the savanna in search of the conditions they prefer. Canopy density and root competition prevent sunlight and nutrients from reaching the understory and create the opportunity for invasive species to outcompete more sensitive grasses and forbs in the savanna. There are few publications on the arthropod species composition of the longleaf pine savanna. We are in the midst of a global population decline. 40% of species are declining at a rate of 2.5% every year. Those statistics are based on what we've been able to track so far, but few studies have looked extensively into the impact of this decline on the longleaf pine ecosystem. Furthermore, a decline in insect populations would cascade through the food web, affecting the ability of amphibians, birds, reptiles, small mammals, and other arthropods from persisting. 70% of plant species in the longleaf pine understory require insects to reproduce. So without them, the conservation of this ecosystem may not be possible. In developing my hypotheses, I looked at 4 key findings from previous studies. Firstly, higher quality plant tissue results in higher insect abundance. Secondly, summer burns yield better recovery than winter burns due to summer being the season of peak productivity for understory plants. So their ability to recuperate from burns is inhibited in other seasons, as summer provides the optimal conditions for growth for the native species in this ecosystem. Thirdly, arthropod abundance increases in blocks neighboring burned sites, as many winged insects take refuge in nearby unburned areas. And finally, disturbance is positively correlated with species richness, which means prescribed burns and shelterwood cutting, which is the type of logging performed in the sandhill gamelands, are likely to increase the abundance of species we see when compared to other undisturbed sites. The purpose of this study is to determine the impact of disturbance regimes on arthropod communities in the North Carolina longleaf pine savanna. My hypotheses are that the site with the most recent burn and no disturbance from logging will have higher arthropod richness overall, that the site with the disturbance will have higher species richness than those without, and that there will not be a significant difference in order diversity and evenness between disturbed and undisturbed sites. I used 3 methods to collect and survey arthropods in order to gain a more comprehensive view of the community. A transect sweep net was used to collect larger arthropods, such as plant bugs and crickets, which are associated with above ground plant biomass, meaning they hang out up in the stems and leaves of the grasses and forbs. A modified leaf blower was used to vacuum a majority of common microinvertebrates from within the foliage. I also used a hand collection to pick up larger macroinvertebrates that are not typically targeted by the other methods used. This is a schematic of the site layout. Each burn block is about 1,000 square meters. Within the site, I selected 2 25 square meter blocks and used a quadrat, pictured on the right, to select 4 square meter plots randomly within the 2 25 square meter blocks. The sweep net was performed separately from the 2 blocks in an area not disturbed by my previous collections. The collections took place in July and August between 11 a.m. and 2 p.m., which are the most active hours for insects in the savanna. The collections were timed, starting with 5 minutes for the hand collection, followed by 30 seconds for the DVAC, and each transect was swept 36 times based on the initial sweep at Site 1. All samples were transferred to plastic bags and stored in a cooler in the field before being moved to the lab freezer upon return to campus. Additionally, I surveyed plant species composition with Alyssa Young as part of her dissertation research and used that data to compare to my arthropod community data. In the lab, all my samples had to be sorted under a stereo microscope in order to separate the microinvertebrates from the other debris. They were sorted initially by order, then by family, and classified further if the specimens were in good condition and did not require DNA analysis to be classified. In total, I had 15 samples, 6 from hand collections, 6 from DVAC, and 3 from SweepNet. Of the 1,028 arthropods collected, 984 could be classified to family, but only 118 were able to be classified to species. On the right is what a sample looks like after all insects have been separated from the debris. I found 132 arthropods in Site 1, which had a burn interval of 2 years and was undisturbed. There were 475 arthropods collected at Site 2, which had a burn interval of 2 years and had disturbance from logging, and 421 arthropods were collected from Site 3, which had a burn interval of 3 years and was undisturbed. The top graph deals with the abundance of invertebrates, or arthropods, and the bottom table shows the number of plant species recorded at each site. There was no statistically significant difference in the number of plant species recorded at each site. In the top left is a sample of Arianae before they are sorted into family. The bottom left photo shows a common jumping spider in the family Salticidae, which is identified by its eye arrangement, for each family of spiders has a unique morphology when it comes to their eyes. In Site 1, there were 14 orders. In Site 2, there were 15. In Site 3, there were 19. The table at the bottom indicates that there was not a statistically significant difference in order diversity between either of the 3 sites, though the difference between Sites 1 and 3 came the closest. In regards to family diversity, Site 1 had 37 families, Site 2 had 73, and Site 3 had 79. The image on the top right shows the family Psychedelidae of the order Hemiptera, which are called leafhoppers. The individual on the bottom is related to those, but it actually falls into a different family called Dictyopheridae, which are plant hoppers, still in the order Hemiptera. The table on the left shows the total number of individuals in each order at each site. 132 individuals composed 14 orders in Site 1, 475 composed 15 orders in Site 2, and 421 composed 19 orders in Site 3. The order composed of the most individuals was Hemiptera, with 276 total collected. The highest peak in the graph of order richness is the 163 Hymenoptera collected from Site 2. It's important to note not only what is present, but what is absent. Coleoptera and Thysanoptera, which are beetles and soil-dwelling thrips, were both absent from Site 1. The table on the bottom depicts the Simpson's Diversity Index for order evenness at each site. Higher Simpson's Index for Site 2 signifies some orders were more abundant than others, which we saw illustrated by the peak in Hymenoptera. In conclusion, my study supported that flying invertebrates are the first to return to burned blocks of the savanna, as indicated by the higher prevalence of Hemiptera and Orthoptera in Site 1, and the absence of ground and soil-dwelling arthropods such as Coleoptera and Thysanoptera. I found that it takes more than 8 weeks for the arthropod community to recover from prescribed burn. Site 2 abundance was elevated in comparison to the other sites, which was likely a result of being a refuge for arthropods seeking to escape the burn. The Simpson's Index showed that there was evenness across arthropod orders and no indication that one was outcompeting others to disrupt community dynamics. I also found that shelter wood cutting, a regime restricted to Site 2 in this study, enabled species richness. Therefore, all 3 of my hypotheses were supported by my results to the extent that they could be, considering a majority of the collected arthropods could not be classified to species. Some critiques I have for this study are that a lot of my specimens were damaged from the collection, so I was not able to classify many of them to their family grouping or further. I was also limited by the quadrats and sampling methods. Many of the photos you see in the field, especially those in this slide, are of invertebrates that I did not survey as part of my study because they weren't within the limits of my quadrat when I was sampling. For future studies, I would suggest that to track correlations between plant and arthropod species to see if there are a coevolution between species in this ecosystem. I would also suggest the use of different sampling techniques to collect and survey adult winged arthropods, which were conspicuously absent from my collections since they focused more on the microinvertebrates in this ecosystem. I would also survey in other seasons to track seasonal variability in arthropod communities. Before I wrap it up, I would like to acknowledge Dr. Sally Kerner, Alyssa, Will, Catherine, and other Kerner Lab members who taught me how to use the methods I used in this research and supported me throughout the whole time. I would also like to thank the Dean of the Honors College, Dr. Omar Ali, for awarding me the Dean's Undergraduate Research Award, which funded my summer fieldwork and allowed me to devote so much time and resources to this study. Thank you. And I will try to stop. Okay.
Yeah, there we go. All right, excellent, excellent job. Yes, thank you. Okay, so we should have a couple minutes if we can spare them for some questions. So does anyone want to start us off with some questions? Okay, I have one. Well, that's a lot of different types of insects. How long did it take you to actually figure out how to figure out what they were? I mean, did you really have to study pictures of them?
I have been doing research in the Kerner Lab for a few years now, and I had been doing insect identification specific to this ecosystem before I started my independent research. So I was familiar with some of the families there already. My biggest resource when I'm identifying is bugguide.net, which is just a comprehensive catalog of all arthropods in North America. And it's just a lot of thumbing through tabs and looking at images and comparing. And it takes a lot of attention to detail to be able to identify what classifies insects into different taxons. I also used iNaturalist as a resource when I was confirming, especially species IDs. I would post it on iNaturalist and get confirmation from other researchers as well.
Okay, that's cool. And we have an overlap between our two presenters with the iNaturalist. So we see how that works. Yeah. Okay, do we have any other questions? Go ahead, Alice.
I'll ask one. Yeah. So I noticed you collected the samples like at 11 o'clock, between 11 and 12. And I don't know, I imagine that, I mean, I just don't know, bugs must have a day cycle. And I know you can't, check everything at once, but would that tend to affect, well, I guess it's kind of a two-part question. Are they most active kind of at that time and overall? And then do they have like a day cycle that's different for different species?
Yes. So my understanding is that it's best to collect between 11 a.m. and 2 p.m. because at 11 a.m. you start to see the dew has dried up, so the insects are more active above ground, and they're starting to fly around and forage. An evening collection might also be aptly timed for insects that emerge towards the nighttime, and that was not part of my project. And would also be something worth considering for follow-ups.
Yeah, interesting. Yeah, so that's something you have to control for sure, though. Thanks.
Mm-hmm.
It looked like fun.
I had fun.
Yeah, I have another question. So, again, you have a whole wide range of insects, and I'm not sure, if it's fair to categorize them into like bad insects and good insects. I mean, I guess they're supposedly good all in their own way. But did you see any changes when, after disturbance or after the burns, like, did it change maybe from, I don't know, some destructive kinds to maybe some healthier kinds for the environment, if that makes sense?
Yeah. I'm definitely going to critique your your use of bad and good. It's like saying bad and good bacteria. That's not effective.
I
know. I would say the biggest difference in what species become prevalent after a burn are those that are able to migrate quickly out and then fly back in. So that's like grasshoppers and crickets, and it's more based on mobility and the forage that becomes available. I mean, even within a week after a burn, you start to see grass and other forbs poking up from the soil. So they're going to come in and just munch away at those fresh snacks. And you'll see less of the good soil arthropods like the Thysanoptera and Coleoptera that we're missing. It takes a while for them to make their way back into the ecosystem, but that's okay because there's not much for them to do at that point because they're going to be breaking down litter and helping with nutrient cycling, which the fire just came in and released all that carbon and all the nitrogen. So they're not really needed yet. And that's the biggest shift in dynamics after the fire.
Okay, thank you. Yeah, very nice. All right, well, I guess we should be closing up our session soon, but we have a last short question that would be okay. And if not, we will finish up. So thank you so much to all of our speakers. You guys did an awesome job, awesome research, awesome presentations. And thank you for sharing that with us. And thank you all for attending and watching us here. So, okay, well, I will officially close the session. Thank you all and have a good day. Happy Friday. Bye.